Hello and welcome to Sam Biology class. Friends, today we shall discuss about reverse genetics. Reverse genetics is a technique in molecular genetics that help us to understand the functions of genes by analyzing the phenotypic effects caused by genetically engineering specific nucleic acid sequences within the genes. It starts with selection of a gene of interest and ends with phenotypic analysis of its disruption. We know the exact sequence of the gene, but we don't know its function. In order to understand the function, first of all, the sequence of the gene is altered by different methods such as site-directed mutagenesis. And this gives rise to a series of mutants. By analyzing the phenotypic changes in the mutants, we can determine the function of the gene. So reverse genetics is based on gene to phenotype approach. Another concept which is related and older than reverse genetics is forward genetics, which starts with selection of a phenotype of interest and ends with identification of the gene that causes the phenotype. So forward genetics is based on phenotype to gene approach. Reverse genetics attempts to connect a genetic sequence with specific effects on the organisms. This technique is used to recover and generate infectious or defective viruses with desired mutation. This allows us to study viruses in vitro and in vivo. In order to alter the sequence of a gene, reverse genetics uses genetic engineering. Several methods that have been developed for reverse genetics are: number 1, directed deletions and point mutations, number 2, gene silencing, and number 3, interference using transgenes. First one is directed deletions and point mutations. Site-directed mutagenesis is a sophisticated technique that can be used to mutate the DNA of known sequence at a specific site. This technique can change either the regulatory region in the promoter of a gene or can make small codon change in the open reading frame in order to identify the amino acid residue which is important for protein function. Alternatively, this technique can be used to create null alleles so that the gene is not functional. For example, deletion of a gene by gene targeting, also known as gene knockout, can be done in some organisms, whereas in another process called as gene knock-in, the endogenous exon is replaced by an altered sequence of interest. Another technique that can alter the sequence of a gene is TILling. This method combines an efficient technique of mutagenesis by using mutagens such as ethyl methane sulfonate with a sensitive screening technique in order to identify point mutations in a gene. Second one is gene silencing. Suppression or interruption of gene expression at transcriptional or translational level is known as gene silencing. Gene silencing by using double-stranded RNA is called RNA interference. In addition, gene expression can also be disrupted by using morpholino oligos. These methods are called as gene knockdown since the effects of these reagents are temporary in contrast to gene knockouts which are permanent. RNA interference can be used to create specific knockout effects without actually mutating the DNA of interest. RNA interference, specifically gene silencing, has become a powerful tool to silence the gene expression and also to identify and analyze the loss of function phenotype. A simple alternative for gene knockdown is morpholino antisense oligos. Morpholinos bind and block access to target mRNA without requiring the activity of cellular proteins and without necessarily accelerating mRNA degradation. Third one is interference using transgene. A molecular genetic approach is the creation of transgenic organisms that overexpress a normal gene of interest. The resultant phenotype may reflect the normal function of the gene. It is also possible to overexpress a mutant gene that interfere with normal gene's function. For example, overexpression of a mutant gene result in the accumulation of not functional proteins that may outcompete for wild-type proteins. And this accumulation of not functional proteins result in the mutant phenotype. Reverse genetics has several applications like gene therapy, vaccine development, cancer therapy, gene editing, and also protein engineering. So friends, this is all about reverse genetics. For more updates, please subscribe my YouTube channel, Sam Biology Class.